Hello, we are going to talk you and presentate you the insulin, about the insulin with our classmates Diana Monserrat Facundo Hernandez and Renata Islas Olmos, and finally me, Alan de Jesus Garcia Yañez.
The insulin is a peptide hormone that is composed of 51 amino acids that has a molecular formula that is carbon 255, nitrogen is 381, nitrogen is 65, oxygen 79, sulfur 6, and it composed by the two popular chains that is the chain A and the chain B. It's going by the two dipole bridge.
Classification according to lipid. Insulin is classified as polypeptide hormone because of its bonds. It is made of two chains, and a chain of 21 amino acids and a B chain of 30 amino acids. These chains are linked together by two disulfide bonds. Additionally, there is a third disulfide bond within the A chain. Number of elements. Insulin is made up of 51 amino acids in total, 21 in the A chain and 30 in the B chain. Although it is not a chemical element of the periodic table, these amino acids are the elements that make up the insulin molecule. Chemical substance. Regarding its molecular structure, insulin is composed of two polypeptide chains, alpha with 21 amino acids and beta with 30 amino acids linked by disulfide bridge. Insulin function. The insulin is a hormone produced for the pancreas. Its main function is to regulate blood glucose levels. When blood glucose levels are high, the pancreas releases insulin to allow the body cells to absorb glucose and use it for energy or store it for future use.
Now, how is this compound obtained? Well, there are two forms of obtain it, naturally and artificial. Naturally is like insulin is produced by the pancreas, a gland located behind the stomach, allowing your body to use your glucose energy for energy, and then that make it enter the bloodstream through the lining of the small intestine. Once glucose in the bloodstream, insulin causes cells throughout the body to absorb the sugar and use it for energy. And the second way that is artificial obtaining, it's like produces by methods based on DNA technologies that recombines the Escherichia coli under controlled conditions to minimize the degree of microbial contamination prior of the release of each batch of bacteria prima. The following test must be carried because the cell protein must contain a host. Also, there is a single chain on biosynthesis that includes the chain A and chain B of insulin linked in each case by an artificial linked peptide. Is also determined by appropriated and validated method and must be also complied by the derived DNA assay of the host cell and the vector. Now, how do we could use this? Well, we have only used one use on of the insulin in one thing, the medicine. The uses and application are that the insulin is only for medical use. It can be used by industrial or domestic use. And in the reduction of the diabetes type 1 and sometimes type 2. So it can be applied to regulate the diabetes problem because diabetes it can makes that your insulin cannot be reproduced on a longer level. So the use of this insulin that they could apply you in the body is to regulate your level and increase the insulin because of this type of diseases, and also generates enough insulin to, well, to maintain the sugar level normals.
Final conclusion. In summary, insulin is a polypeptide hormone crucial for the regulation of blood glucose levels. Its complex molecular structure, formed by two chains of amino acids linked by disulfide bridges, categorizes it as a notable chemical substance. Furthermore, it is very important to highlight that insulin is a chemical compound with a specific molecular formula, consistent, consisting of 51 amino acids in total, divided into chains. Chain A with 21 amino acids and chain B with 30 amino acids. These chains are linked by disulfide bridges, which determines its classification of a polypeptide hormone.